Welcome, everyone, to this episode of the NLN podcast, Nursing Edge Unscripted, the Scholarship Track. I'm Dr. Sue Forneris, and I am the Director of the Division for Innovation and Education Excellence at the National League for Nursing in Washington, D.C. Our unscripted conversation track entitled Scholarship celebrates the published work of select nurse educators from the NLN's official journal, Nursing Education Perspectives. In these conversations, we're going to embrace the author's unique perspectives on teaching and learning innovations and the implications these have for our nursing program development and enhancement. In this episode today, we entitle it Exploring Perspectives on Vaccine Development, and we'll focus on the unique perspectives of Dr. Karen Frith, who is the NLN Nursing Education Perspectives Emerging Technologies Editor. Dr. Frith is also Professor and Associate Dean for Graduate Programs at the University of Alabama College of Nursing in Huntsville, Alabama. So welcome, Dr. Frith.
Thank you so much.
So your particular column article was featured in the July-August 2020 NEP publication, and it was entitled 20-Year Synthetic Biology Research Roadmap, Implications for Vaccine Development and Future Research. And it came out at a really timely, point in our own trajectory here as citizens of the United States and what we're going through with this COVID vaccine pandemic. So tell me a little bit about what made you decide to do a column in NEP focused on this.
So I work at the University of Alabama in Huntsville, which is a university that is highly focused on science and technology. We are located 1 mile away from NASA Marshall Space Flight Center and from the Missile Command Center in Huntsville, Alabama. So it is a very unique place. And when I do my research, we work with others in engineering, science, computer science, because we're the only the only health science program in our university. So we're not associated with an academic medical center, and it's a small university. It has 10,000 students in it, but it is classified as a high research institution because of our work in sciences and engineering.
Yeah.
So when I came to the Huntsville area and began working in research, my natural inclination was to reach out to those in computer science. I have a background in informatics, and so it has been the most fun working there. But because of those associations with my colleagues, I read widely in the engineering and science literature.
Yeah, and that's not an area when you think about academic health centers that you hear a lot of real direct connection between nursing programs and the engineering departments
Right.
and computer science. So that's really a valuable intersection.
It is. And so in 2019, the 20-year synthetic biology roadmap was published. And so I read the piece, and it's a highly technical document, and it was developed by over 80 scientists across the world. And in that roadmap, it really talked about the various areas that could be influenced by this work of engineering and biology, or engineering and health. And so one of those areas was in vaccine development. Infectious diseases, but also non-infectious diseases. So I remember sitting at a conference in January 2020 and seeing the first map that came out in Johns Hopkins website tracking the coronavirus vaccine, and thinking, and I remember reading the document in 2019, and I thought, oh my gosh, this is the time. This is about to become a really big problem for us. So I went back and I found, again, the document, reread it, and it stunned me. I thought, this is what's going to be the pathway for us, because I knew that flu vaccines take far too long to produce a vaccine that's effective. So there had to be some new technology out there. And so that's what really gave me the thought to start on it.
Yeah, it was timely. It was really a timely, excuse me for interrupting, piece, because, to open up the journal in July, August, and to see that on there in the midst of, the vaccine on the thrust of being able to be released, I thought, wow, what a timely piece. There was a sentence in there are a couple of sentences that were very intriguing to me as you read through it, but there was a particular citation from Founds in 2009, and it was interpreted by you as nurse scientists, particularly those in programs of research that focus on system biology, crucial members of these engineering biology research teams. And so I thought, gosh, you must have some knowledge or thoughts about examples of the work of nurse scientists that are doing this work.
Yeah, so we at the University of Alabama in Huntsville have several of our nurse scientists that are working in this area. For example, my own research is with a computer engineer, Dr. Amelia Vonov, and we've been working together for 10 years on digital healthcare. While it's not genetic based, it is based on personalized medicine. And what we're trying to do is to find early biomarkers of mobility problems so that we don't wait until someone falls to begin those interventions. And so there, of course, we have smartphones, smartwatches, and other wearable technologies. And so that's an area where I'm focused. A couple of my other colleagues at the university are focused on genetic research with others. So Dr. Louise O'Keefe did a study with Gene Capture, which is a biotech company here in Huntsville, and her work was on working with this company to test a device that would take small samples of urine and detect the bacterial infection within 45 minutes, whereas a culture takes over 24 to 48 hours. And that research study was successful, and they did find that there was a 90% validity and reliability of those studies, and so of the urine samples. So that's another application that is fascinating and could change bedside diagnostics to give people the right antibiotics at the time they need them. So so just awesome.
A quicker way, yeah. Wow.
And then, Dr. Susan Alexander is working with our atmospheric science team here in Huntsville. And so they use sensors in the environment, and she correlates the atmospheric readings with the incidence of cardiac and pulmonary disease that results in hospital visits and in ER visits. So there's just a world of opportunity. One of our new PhD students is working with Dr. Pam O'Neill and Dr. Elise Adams, and she's looking at the gut biome of neonates and trying to see, is there an association with colic? Now, all the mothers, if this work can be successful and you can figure out, well, what is a biome in a infant's intestinal tract that is good versus one that leads to colic, that's going to be a major contribution to motherhood.
Especially firstborns.
Yes.
When you make the decision whether you want to go through this again or not. So
absolutely. And of course, there are other universities out there. I just highlight our own researchers because I know their work so well. But Duke University has a very fine program, and there are many others that do as well. And so it's not, while it might seem in the distance for nurse scientists, it's really not. It's here.
Yeah, that's just amazing. The far reaches of the discipline in terms of its contribution in healthcare.
Right.
Absolutely. You mentioned your own work in biomarkers, and I know that there was some areas of the article that talked about the automation technologies and the fast sequencing.
Right.
And there was a term that was used, synthetic biology, and I'm wondering if you can talk a little bit more about the COVID-19 vaccine in relationship. Is this a synthetic biology and this whole notion of biomarkers?
Yeah, so it is. It is definitely, and synthetic means engineered. So if you think about bioengineering or biomedical engineering, this is the area of the intersection with our health concerns.
Okay.
And so the way that the messenger RNA works is that it introduces a molecule that stimulates the production of an antigen, and then it just disappears from the body. It's a messenger RNA, so it doesn't infect the host. That would be us. And once it causes the protein to develop, then there is no other purpose for it, and it just dissipates from the body. So it's quite it's safer than most other kinds of vaccines. Now I say that, and we all know that right now we're in large data collection at this point, and we won't know for many years really the long-term effects. We know short-term it's very safe.
Right, exactly.
Yeah.
And I know when you mentioned the use of the RNA vaccines, that their use in manufacturing these vaccines, or the discovery of these vaccines, is the technology is faster.
Right.
And that's been somewhat of a media speculation, so to speak, where the public is concerned about this use of media's fast tracking of vaccine preparation, and perhaps you can speak to that a bit, because it doesn't necessarily mean it was done differently than any other preparation, and I'm wondering if you have some thoughts.
I do. Thank you for that question. So I wanted to start by saying that the whole idea of using messenger RNA as a method for vaccination goes back about 40 years. And so the science has been around for a long time, but the ability to use that science in large scale, at large scale purposes, is just now coming to the forefront. And that's because gene sequencing now has gotten rapid, just like the example that I talked about earlier on the urine samples that were tested for its genomic structures to diagnose the presence of bacteria. All of this work now is made fast through very rapid analysis of genes and genomic structures because of the computing power that we have now. And we didn't have it years ago. And so the progression of the technology, really there are a couple of points in time. So 40 years ago, and then in the early 2000s, there was some work done that looked like, okay, this might be possible. But then it was in 2017 when there was a nationwide, you could start seeing the movement in this area of RNA vaccines and really engineered solutions. And there was a chapter written by Cramps and Elvers it was called The Introduction of RNA Vaccines in 2017. And then there were a lot of publications in the scientific literature that started in 2018, just in time for the pandemic.
That
And they didn't know about it, of course, but they knew that there are other illnesses like Zika and Ebola that are all viruses as well. And the idea that there would be another pandemic is widely known in the medical literature. We've been just waiting on it, and it has come. And so the scientists that were working on it, while it's fast-tracked in the media, it has not been a fast track in the scientific literature.
Just the technology has so much more progressive at this moment in time, and we haven't had a pandemic,
right?
Essentially.
Right.
And so that does make sense. So would you say then that, as you think about that, in terms of setting a benchmark for future vaccine, is this COVID-19 vaccine production and the whole science behind how that evolved, is that a new benchmark for us?
I think it is. It's a new method, completely different from the other types of methods that have been used with live or attenuated viruses. This does not contain any virus at all. And so it's just a completely different method. And one of the things that it will also influence, and the pandemic, as we know, has had terrible, terrible effects on the people's lives, from health and economy and lost jobs and those kinds of things. And at the same time, there have been innovations that have come from it. And this is one of those. And so it's not only for infectious diseases, but RNA vaccines can be used and are being researched as solutions to cancer. So there are probably 1,000 clinical trials going on right now. Some of the ones that are probably the most promising are melanoma, and of course that one is a very aggressive cancer. And so this work on RNA vaccines for this virus will probably accelerate the work that can be done to find real solutions to cancer treatment.
Oh, that's fascinating. One of the other pieces that this also gets to is chronicity, as we look at some of the chronic illnesses, of our own time and nurses' work in that. And that was actually part of a strategic plan that was mentioned in your article that came from the Institute of Nursing Research, this personalized health and the future direction of nursing research, the practice of nursing research. So, you address this really important point, and that has implications for teaching and learning with our graduate level preparation of nurses in our discipline. And so, as you think about our undergraduate and looking at a trajectory of practice moving forward, talk to me a little bit about how you think this informs preparation as they begin, their part in the practice.
Okay, thank you. Great question. So one of the things that I think about with personalized health or personalized medicine is that if everything continues as it looks like it will, and it's successful, this type of work is going to be as groundbreaking as antibiotics work, so that we won't talk about people dying from certain diseases in the future, because there will be this genetic work that will help either make it a chronic disease and not cause mortality. And so we need to educate our nursing students to be prepared for that. So one of the first things is, while we're learning about RNA vaccines, our nursing students need to learn about them too. Because one of the things that we know is our students who are in clinical practice with their faculty members are always asked by patients, well, what do you think about this, that, or the other? The patients don't really know that they're a student. I mean, they know, but they think, oh, well, she's a nursing or he's a nursing student. So they ask kind of hard questions. So we have to prepare our students to be able to answer questions about the safety and efficacy of RNA vaccines. And we have to teach them about possible new ways that they could be delivered. So we all think about a vaccine as a needle in the arm. But of course, we know there are other ways that you can deliver vaccine. You can deliver it as a spray in the nasal cavity, and there are different ways. But one of the new interesting ones is a vaccine delivered on something that looks like a Band-Aid, and it has multiple arrays. And what that means is small microneedles on the shape of a Band-Aid, and it's pressed into the skin. And then after the medication is delivered, then those microneedles dissolve because they're made of sugar and other kinds of dissolvable substances. So can you imagine that we have a student who goes to make a home visit, and with the home health nurse delivers a vaccine by just pressing a Band-Aid against the skin? And so we have to teach them about that and why
Well,
that.
the patches, when the patches came out in terms, so it's very similar in some ways, maybe a little bit higher a level of explanation for the patient, but yeah, absolutely.
Yeah. I also wanted to think about, I mean, we also have to prepare our students for sensor-based health. There's a lot of literature out there leading me to believe, and many others, because a lot of research is being done in it, that our sensor-based wearable and environmental sensors may actually provide real-time data, or at least collected data by healthcare providers that can be used to assess people in their daily lives, not just in an office visit. And so we need to prepare our nursing students for that kind of technology. And you pair those kinds of wearable environmental sensors with information about the genetic makeup of an individual, we're talking a whole new world of healthcare.
It's funny that the conversation continues, because this whole informatics and use of data to inform practice trends, as you think about, especially as we know, nursing practice will become more and more community-based with, sicker and sicker people in the hospital, changing sort of the dynamics of the volume of people in hospitals versus the volume of care delivered, in the community. I've often, made the comparison that as a practicing ICU nurse back in the day, those patients now are seen in today on general med surg patients, and the patients in ICUs today were dead, 40 years ago. So it just, it's going to be a different kind of nursing, I think, as we move forward. And this information is really vital and as we prepare.
Right. And I think about when I was teaching in undergraduate education about diseases like asthma. And now we have to think about how we would teach students about using air quality sensors and pollen level, and how we would look at inhaler uses and adherence measurement, that all of those data can be pulled together. And instead of teaching them strictly about how a medical diagnosis is made, and it really moves more into treatment and how a nurse is central in the treatment of that. And so other kinds of technologies that can be used are breath nitric oxide levels. So exhaled nitric oxide, which is a very individual measurement and in daily life could be used. And so you put that with a geosensor, air quality, pollen levels, and we get to a place where not only are we treating, but we're predicting, and we're using these preemptive plans to keep people from having an asthma attack.
Right.
And nurses will be at the center of that.
Yeah, trying to curb the behavior, trying to reinforce health practices that would move
Mhm.
them, yeah, down a different trajectory.
Right.
Absolutely.
Yeah.
Oh, this has been so fascinating, Karen. I appreciate your expertise in this area, and we look forward to more that is going to come as you continue to be the editor of the Emerging Technologies column. Lots of really good information there, and to our nurse educators as they work with our future professional nurses. So,
Well,
well, thank you for joining me today.
yes.
I really, really appreciate it. And for all of you who are interested in reading this column, you can go to Nursing Education Perspectives, and it is the July-August 2020 edition that you'll find Dr. Frith's column. Thank you.
Thank you.